I'm Ann Estes from Biological Sciences, and I appreciate you giving me a chance today to talk about my entertaining ways to assess students. We are living in a microbial world, and I am a microbial girl. Yes, that is Madonna, and I teach microbiology. So I often try to find fun ways to do it and to assess it, because I think of assessments generally as a very stressful thing and an overwhelming thing, and students find it that way too. So I try to find different ways to do it. So instead, we're saying, yay, we've got that done, we learned something, and maybe we had fun too. So the different things I'll talk to you about today are a pre-exam bonus, and exam alternative questions, the lab practical poster, and I won't talk about my microbes in the media today, but I'm happy to talk about that to you individually. It's a written assignment, one page, that compares the media article to the science article written, that the media article is written about. So the first is the bonus activities. A minute sketch with folded list is one thing I like to try to teach them as a new way to learn. And the idea is to make them practice recalling information while chunking the information in the context of learning objectives. So then they go, yay, I figured this out, I synthesize things. And it is an alternative study technique. Again, forcing them to recall, forcing them to synthesize things, and really find the forest and get out of the weeds. So it also forces them to use arts and science together. I think science is a very creative process, and different memory research has shown the more types of senses you use helps you remember information better, as well as, of course, repetition. So I try to get students to use visual, kinetic, and auditory methods to remember this information and process it. It teaches me their perspective, kind of where they are before the exam, so I can try and address problems. It also gives me their pop culture references, so I don't reference a 20-year-old Madonna song. It makes them recognize what they do and do not know when they have to recall this stuff and synthesize it this way. And it also teaches them how to teach, which I think is a good learning mechanism. Finally, I'm a big science communication advocate and have many social media venues I like to engage the public at. And so if my students' resources that they generate are really good, I like to share them on my social media sites so that we're educating the public, we're empowering the students, showing them, hey, they really did get this information right, they synthesized it, and now we can use it to educate others. They can share it on their social media networks, which are different from mine, and so we're educating more people. So this is kind of a side win in some cases. So here for the folded list and minute sketch, we've got a learning objective of humans or microbial worlds. So we did this together in class. I do this several times in class to kind of teach them how to do this. And so we came up with this list of words: microbes, microbiome, birth, niche or habitats, disturbance events, selection, and planet us. And I focused on birth and niche and habitats. Took literally a minute to do this drawing. And as my drawing prowess shows, you don't have to be a good artist. You can use simple shapes to represent these things. After all, they are bacteria. And so here we have our infant, child, and adult. You notice there's circle bacteria only associated with the head. With the feet, we've got these squiggles, and then the guts, there's different shapes, except for in the baby. There's only one type of bacteria, this star type, that we see a little bit in the adults and the child, but they've got others too. So this kind of gets across the idea that different microbes are living in different places on us as microbial worlds, and it's due to the food, oxygen, and temperature of these different environments. So the next bonus opportunity I give the students is a social media summary, where we're using a single image to summarize a microbiology concept in a fun way. And our learning objective in this one is that microorganisms and their environment interact with and modify each other. So I saw this great cartoon done by a person I follow on Twitter, and here's the social media post I would write to go with it. The Earth's early atmosphere lacked oxygen until cyanobacteria came along. Cyanobacteria do photosynthesis, giving off farts, of oxygen. This changed Earth forever. Oxygen-sensitive microbes died or had to find anaerobic habitats. This is often called the Great Oxidation Event. And so here's our anaerobe who's saying, dude, your farts are lethal. And of course, I have hashtags to engage the public. So, this would be an example of how to do a social media summary. Here's one from a student. Another Cinderella story, the bacterial edition. Here's Cinderella in her ashen clothes looking sad. The fairy godmother says, "Oh my dear, we can't have you going to the ball looking like that. Bippity-boppity-boo." And some of her DNA flies over and around Cinderella, is incorporated into Cinderella's genome. And now she has this beautiful ball dress. Her hair's all done. "Oh my, it's beautiful." And the fairy godmother says, "Now that is what you call a natural transformation. You're a newly transformed bacteria on the inside and outside," which is what our learning objective is. Natural transformation is where they can pick up DNA from the environment, and now they have a new trait. So I can take this into class the next day, and I can continue class discussions and review this way and say, "Okay, class, so we know that bacteria don't have a fairy godmother. What makes them naturally competent? And, what allows natural transformation to occur?" And we can talk about it. So it's kind of a fun jumping off point. And it's generated by one of their classmates. So another example is memes. Students like to do memes. This is talking about different cell walls. And so mycobacteria is saying, "Murine in your cell wall can't protect you against disinfectants as well as mycolic acids in mine can, but that's none of my business." And then she's got it annotated more in the rest. I made a meme to kind of get across to students how to properly write a scientific name. And this is Drake, a singer that some of the students said they liked, so that's why I had Drake saying, "Don't do it this way, do it this way instead." So hopefully that'll help. The way I assess this is to make sure that they've identified that learning objective and then visually represented it scientifically correctly. They cite any images that are used, and if appropriate, they're using hashtags, word lists, corrections. So this minute list and minute sketch and folded list is something they should actually do once, and then they should go back to their notes and see what they've missed and add things in as appropriate, make a new list, and then make a new drawing from that new list. So that's where I would expect corrections to come in. I give them 2 opportunities to do this for a total of 20 bonus points out of 900. So it doesn't make that much of a difference. But if they actually learn these new techniques, some of the students I've had go from making F's on the first test to an A in the class after they've incorporated these techniques. So I think they can be pretty powerful. Another thing that I really formalized more during the spring pandemic were these exam alternative questions with microbial movies and microbial poetry, because students had said they were kind of feeling down and stressed out. So I thought this might be fun. So usually I give them a 15-point question where they're trying to talk about different organisms and how they have different characteristics to allow them to live in very different environments, from like the Arctic all the way down to, your gut. So instead, I told students they could do a movie. And here's an example of what Nico did.
So I'm going to do a little bit of like a draw my life, except it's going to be about bacillus bacteria and how they can do this cool thing called sporulating. And so bacillus, when they have low nutrients, that's when they start to form spores. And they do this by this kind of whack but cool process where they make a copy of their DNA, they form a septum in their cytoplasm, and then form a membrane around that immature spore, and then release it as a mature spore.
So this was cool to me because, number one, I didn't know about Draw Your Life, which was a big social media thing evidently about 10 years ago. So that was fun. And the other thing is he actually incorporated some information from a peer-reviewed literature paper I hadn't read. And so I learned something, and that was even better. So I loved the job Nico did on this. So this was the rubric I had given them. I kind of give them some different ideas of style, but really focused on the structure that they needed to introduce their microbe, explain what it does that's so unique, and then summarize this whole thing. It had to be under 2.5 minutes, no jargon, credits, captions are ideal. I gave them a lot of resources to use, though, both students kind of did their own thing, which was even more fun to see. And I gave them several examples so that they would have enough structure to kind of know how to do this. And I gave them a little bit more time than pass the exam, since it does take a little bit of time to make these creative things. But both of the ones that were turned in were really great. So that was exciting. When I taught this past spring, it was April, and that's National Poetry Month. So I decided to give the students an opportunity to write a microbial poem that illustrates a point about the material in exam 2. So examples of these, I gave them examples of what an acrostic is, a haiku, a simile, or a free-form poetry, and they could choose any of these. This isn't something they can do on Chegg. So here's one that I got that was really fun. This is an acrostic. So if you look at the first letters of each word, it spells out salt. And here's the poem. Special sodium, potassium pumps, and aquaporins let halophils thrive. This is by Alexis, and I annotated it. Halophiles are these salt-loving bacteria. That's why the water here is pink. And here's some of the different structures she's talking about. So that was fun. Here's a haiku from Avanti. Those heat shock proteins are good habitat blessings for thermophiles. And finally, another acrostic that I really loved. Microbes manage everywhere in human soils and ocean depths. Cultured to find out more while rising with dust and falling with rain. Our microbes meet where we have met, beyond what the eyes can see. Everywhere to help with life on Earth. So let's see where it takes me. I just love that. I thought that was really fun. You might notice that these last two things say created in BioRender. This is a program I use a lot. It has scientific clip art that you can use and make into whatever graphics you want. And it's a really powerful tool, free tool. You can do 5 different things. So I've been annotating the students' poetry this summer. The last thing I do is a lab practical poster. As a student, going into practicals, you have stations set up where there's a different question at each station. And often, as both the teacher and student have noticed, people just dump the information afterwards. So I wanted to make them work through the information more, to think through it. So the goal here was having students work through the methods to identify different bacterial species and distinguish between each species. The structure I gave them, I provided them with the different microbiology tests they could use and the different organisms that they could look at and compare. I gave them flexibility on kind of how to present it and gave them a lot of resources to use. And here's what I got. So here's 2 different ones. I did let them work in pairs on this because I thought it'd be good for them to troubleshoot this and bounce things back and forth, and they did that really nicely. But in both cases, what they're doing is talking about the different kinds of tests and then how those test results differ depending on the organisms they use. So it was really successful and much more interesting to grade. So I hope you've enjoyed these different ways to assess in maybe a more entertaining way. And of course, I'm happy to talk about this kind of stuff and would love to hear your ideas too. You can reach out to me on email at astis@towson.edu. You can also find me online at Mostly Microbes on Twitter and Mostly Microbes host on Instagram, or my science blog, mostlymicrobes.com, where I have some more ideas and examples of this stuff. And here's an example of something I did in BioRender to send the students again after the survey, where they would tell me how lonely and depressed they were. I put a bunch of microbes on it and said, even in quarantine, you're never alone, because we always have our microbial friends. So thanks again for this opportunity, and I'd love to hear what you're doing.